electrical engineer at Bell Laboratories. Anyone here at Bell Laboratories? It's amazing to say, I can go into my classes and I can say, anyone here at Bell Labs? And these 22-year-olds say, who? So, and if I tell them they invented the telephone, they don't get it. But over those 35 years, I became fascinated with the question of how do you translate technology into impact in society? NSF spends how much a year in research funding? Anyone know that number? $8 billion. How much does NIH spend? 32 billion. That's over $40 billion going into research. And yet the impact on society of that $40 billion is a big drop off. And the NSF recognizes, they created a whole new director around it, but it's known as the Valley of Death. And it's always fascinated me. I saw this at Bell Labs as well. I worked at Mass General for 2 years in anesthesia research. I also did research out in the West Coast, another school out there that does research in that area. And the thing that always got me was great ideas in research, just were not making it to market. And so 6 years ago, when I was asked if I would teach a class at Tufts and then also launch a program, I thought to myself, what a wonderful opportunity to create a program where we could bring together researchers, people like Xiaobing, with STEM graduates, recent people who have that strong technology background, but who really want to do more with their life than just the research part. That's an important part, but to get it across that chasm, you really need to find what is the real problem you're solving? Who's the customer? Your classic questions, value propositions. What's the business model that's scalable and also sustainable? What's the financial model to make it investable? And most important of all, what are the people skills that you need to be able to persuade others to come along your mission, raise the money, and get things done with raising a team? And in 1 year, I've had the honor of being able to attract people like Lara, like Konstantinos, like Gene. You're going to meet some of these people in these presentations who had that passion. And in that 1 year, we could use it as an opportunity to incubate ideas. So you'll see examples of that. But I also wanted to say one other thing. Another part of the magic is bringing them together with the community. And so Michael Singer's been a good friend of us from the very early days. But the value is connecting people like Michael with people with the students and with the broader community here. So I want to encourage all of you here with your hats on. I mean, that's how I met mine this way as well. The broader we can cast this net, the more we can create that connectivity, the higher the probability that the serendipitous connections get made and things will blossom. I think I was talking to Brendan, he's out of San Diego. If you look at the trajectory of the life science startup industry in Boston, it wasn't always like this. 20 years ago, it was very nascent. But I think it's because the community got together. There's a little bit of seed capital coming from the state to kind of kick it off. But the community now is a self-sustaining machine because of the strength of the community. So I would welcome an opportunity selfishly to any of you interested in interacting with students at Tufts. I'm always happy to bring them in. But more importantly, I really thank all of you for supporting this kind of event, because I think this is how we build a stronger community. So thank you very much, all.
Thank you, Kevin. And now I think it's time for us to introduce our very first company, Anavet Nanotech. And we have here tonight Konstantinos Georgakis, who's the Chief Business Officer, who will be leading the discussion.
Hi, everybody. Good evening, and thank you very much, Laura. Great being here, and thank you very much for the invitation. So, outside. Yeah. So at Anodyne, we are developing new forms of existing drug products to replace painful injections with convenient transdermal patches that can be self-administered at home. We are doing that by leveraging our patented microneedle technology that has the ability to deliver milligrams of biologics transdermally. The reason we are targeting biologics is very simple. There are no alternatives. So these are very large molecules that cannot pass through the skin. And they also have very low bioavailability, which makes oral delivery even more challenging. Bioavailability when delivered orally, sorry, which make oral formulation more challenging. So they require injections. And there is a single problem that causes both patients to suffer, but also pharmaceuticals to miss out on revenues. And these are problems with injections. Injection concerns like stigma, mental and physical discomfort often leads to delayed initiation of treatments, as well as decreased patient compliance. And as a result, pharmaceuticals are missing out on billions of dollars of revenue every year. But most importantly, patients do not receive the right treatment, so they are suffering. What we propose is a very simple solution. An easy to use, self-administered patch that does not require any cold chain distribution or refrigeration. To replace existing injections that are currently delivered daily or weekly. And to achieve that, as I mentioned, we have developed a new class of microneedles that are solid-state porous polymeric microneedles. So, as you can see here in the picture, we incorporate the dry formulation of the drug throughout the whole structure of the needle. And this way, we are leveraging all the available volume. And this gives us the advantage that we can load much more drug and deliver much more drug than anybody else can do with existing technologies. And by the time we apply the pads in the skin, the interstitial fluid, which is the surrounding fluid, will extract and dissolve the desired API, the drug, and will leave behind an empty matrix of the microneedles. And then you remove it all away. So a little bit of our story, how we got here and where we are today. So we started back in 2019. We're three co-founders, Jay, Josat, and myself. We all met at Tufts, as most of the companies that I've seen today. Jake and I were doing the MSIEM program, where we had the privilege of working with Kevin Noe, Laura, and other great people there. And Josat was the lead researcher and the inventor of the technology, was finishing up his postdoc there. We also have a fourth member of our team here, Michael Singer, who was our first investor, advisor, and now director as well. The next 2 years, from 2020 and 2021, we proved the technology, but also validated the commercial need. So to prove the technology, we delivered through our system a large amount of the monoclonal antibody rituximab in non-human primates, as well as achieved successful pharmacodynamic effects in rodents using the peptide, and provided the chemical stability required for at least 8 weeks. At the same time, we were able to secure 2 early collaborations with top 20 pharmaceuticals and raise $4.3 million from private investors. Right now, we have a great team of 9 full-time employees and we are all of us are working through establishing our GMP process to move towards our first clinical trial that we plan on doing in 2023. At the same time, as every startup, we are raising money, so we're raising our Series A, which is going to be for $10 million. And we are always discussing new potential opportunities for collaboration with other pharmaceuticals. That's it. Thank you very much. If you have any ideas, if you're interested in more, or if you have any suggestions, I would love to connect with you.
Yeah, great presentation, and yeah, we'd like to take some time to have any questions from either any of the panelists we have or any of the audience members that have come up. Yeah, we'll start there.
Thank you. How long do you need to keep the microneedles within the surface of the skin in order for the drug to diffuse?
That's a great question. So I would start by saying that it's tunable, but for now we usually leave it for about 24 hours for our preclinical work, but the intended use is much shorter. So we envision this as an overnight patch at least.
Yeah, right there.
Yeah, thanks, Konstantinos. From your animal studies, do you have pharmacokinetics or even efficacy like receptor binding data, something like that, to give us promise that this will behave like an injection?
We do have all of that. I cannot share because most of the data is confidential, But I can say that it will mimic the subcutaneous profile with an expected, as every transdermal system, some delay, but much more sustained pharmacokinetic.
Thank you for the presentation. You may have just answered my question, but what methods of delivery will this replace? Is it purely for sub-Q, or are there other injections you can replace?
That's a great question. So right now, the reason we are focused on subcutaneous is because the feedback that we initially received from pharmaceuticals is that they much rather replace something one by one, meaning that if it's something weekly, just replace with a weekly. If it's subcutaneous weekly, just replace it with a weekly patch, or if it's daily, just with a daily. So we didn't want to go more on the switching from IV or something else. But right now we have many pharmaceuticals that are actually reaching out to us, like the last year, for completely different molecules. Some are orally delivered now, some are also IV.
Great. Yeah, over here.
Yeah, I've got a question. With biologics, the immune system with humans and generate immune response. Just wondering, you can study if you've gone through the skin, potentially replace it.
You mean more the topical effect, or
Well, in the skin,
right.
you can potentially, the immune system recognizes the skin, the body, it's like human responses.
I can also ask for help from Mike here from this one. I also have an answer, but I'm curious to see yours.
I was in the school district when I was giving a talk at Ryan's Convention Center. Our chief medical officer was in the audience. I did the same thing. I'm a physician by training and developed a few monoclonal antibodies for Novartis. So yes, this is a concern that the company's thought about, and I think we'll have to be empiric to see if it's true. But as you know, with patients injecting subcutaneous antibodies at home, inevitably some of that antibody does get into the skin or subcutaneous tissue. Patients don't have perfect technique. Doctors don't either sometimes. So we'll just have to see.
Thank you, Mike.
Sure. I know some other people are working on like oral biologics, especially via some transcytosis and glial cells in the gut. How is your efficacy comparing to oral biologics that's currently?
So the only oral biologic that is currently approved is from Novo Nordisk and semaglutide, and as you can see, the bioavailability is, I think, like less than 1%. So something that is weekly injection is now like twice daily, and we've talked to many physicians and they don't prescribe because it creates like side effects and people are like, getting confused. So as of what is currently delivered, it will be much currently approved. It will be much better because we can do one by one replacement. But there are also like many other researchers, investors, startups that also develop new formulations. So I don't have the exact answer.
So how much can you load and how long does it take? It's a diffusion process, right?
Correct.
So how long does it take and how much antibody can you
So we can load about 5 milligrams of biologics in a square centimeter. And I'm saying square centimeter because it's a very tunable process, so we can make pretty much like any patch that can be commercially viable, so it shouldn't be like too big. And how much does it take to diffuse? So we usually leave it on for, as I said, like 24 hours for animal studies, and all of the API will diffuse. But it also, in vitro, it's much faster, so we expect like a very immediate release.
Yeah, I want to wrap up with a kind of non-scientific question. So was there a moment, or what was one of the moments where you and your team knew you were really onto something? And, was there like some sort of, time where you can go back to and say, this is when we knew we had an incredible invention we were building?
Also a great question. I need to think about it. So
I have the answer.
Okay.
When they were in my class.
Exactly. Yeah, so mine more. It was when we were still at TAF. We had some great traction. We won some competition money, but as you know, it's very different the student entrepreneurship that when transitioning to a real company. So when we started raising money and like when we met Mike and other great investors, we saw like we have some great traction there. That's where we realized that it's something worth pursuing.
Perfect. Let's give a round of applause. And yeah, thank you to Anodyne Tech and everyone for all the great questions here. And yeah, really looking forward to seeing the progress and you know do some updates of some of the future events here. So yeah, with that, really excited to welcome the next company, Cellens, who's represented by Jean Pham, to come out and present for us tonight. And yeah, excited to welcome us.
Thank you, Jason.
Thank you. Yeah, you can take it away.
Hello, everyone. Thanks so much for having me here. I would like to start just by giving a special shout out for the organization. And as you know, Boston New Tech is collaborating with a very special nonprofit. It is called Viet Challenge. And a special thing about this organization is that during the day, you would see us as corporate lawyer, as engineer, scientist, and startup founder like myself, but at night you would find us working on Zoom and organizing things like this. So if you really like this kind of event, I hope you would come back and support it next time. So thank you for coming here today. And now, yeah, I would like to share about what I'm doing with Cellens. So yeah, at Cellens, our vision is to bring clarity and precision to cancer diagnostics. We envision a world where it's not painful to detect cancer. And I would like to start with a very interesting fact about cancer that some of you may not know, which is the fact that cancer cells actually feel different to the touch. Before the age of pathological imaging or other modality, people actually rely on touching a mass to see if it were malignant. And labs around the world, not just in our lab, has shown that cancer cells actually feel much softer than morphologically similar non-cancer hurt cell. And this insight actually helps us to learn much more about cancer cell beyond what is currently available with other molecular diagnostics. So at Cellens, we're using a mechanical tool called atomic force microscopy. It's kind of like using a hand, touching a surface, and then feeling the softness, the adhesion, height, and other physical property on the cell surface. So we're calling our cell as using a physical approach to detecting cancer. We have paving the way to use cell surface property as a diagnostic marker. We have done some proof of concept study in our lab to show that we can use different cancer types, like ranging from cervical cancer, colorectal cancer, and bladder cancer to detect not only cancer, but even to differentiate platform. For today, I wanted to laser focus on our first commercial application, which is a monitoring test for bladder cancer. And the reasons, as you can see here, is because the current standard of care for how patient gets monitored with bladder cancer is so painful, inconvenient, and expensive. Nearly 1 million people in the US right now showing up at the urologist with blood in the urine, and they have to do a cystoscopy, basically putting endoscope into the urethra to visually see where's the tumor lesion. But it doesn't stop there. After the initial treatment, because of the high recurrent rate, 50 to 80% chance of the time, they would have to go back to the urologist every 3 months, 6 months, or 12 months to get regular checkup. So this is a big problem. And this is where Cellens come in. We have interviewed more than 100 urologists to realize the clinical unmet needs in the monitoring market, where we can help urologists better manage their patient. And our IP method is revolve around the fact that we can use cell from bodily fluid, and in this case, from just a urine sample. So pee in a cup, we get the cell. We have a proprietary way of preparing and preserving the cell surface, UAFM, and then machine learning to get a diagnostic score of having cancer or not. We have done another trial with 68 patients at Dartmouth and UW Seattle. 23 of them have active cancer at the time, and we were able to show a 94% overall accuracy, significantly higher than cystoscopy of just 74%. We have 91% sensitivity, 82% specificity, and great area under the curve. So this is a really promising result, and we had been partnering from day one with clinicians to develop clinical guidelines, because we strongly believe that there is a need to have a better way to manage bladder cancer patients. And we are very thankful to have the support from top-notch urologists at these medical centers. Looking at the bladder cancer market a little bit, there is nearly 1 million people right now with bladder cancer. If they do 2 tests per year, that is 1.6 million in testing opportunity. And at the price point of $700, that is 1.1 billion annually in testing opportunity. So this is an initial market, and it's already 1 billion. And this is where we are right now. I'm very proud of the team. We have a small team, but we've done a lot. We have been in clinical validation with 5 hospitals right now, with 3 different types of state to predict the aggressiveness of cancer. Nearly 1,000 people right now are enrolling in our clinical validation. And the last point, which is the reimbursement pathway, there actually are the commercially available tests on the market, like Eurovision or CX Bladder, but their accuracy rate has not been at the level where urologists would feel comfortable using, or because it's a genetic test. One they order and half the test, they don't need it for a second time. So we solving both problems for urologists. Currently, as I say, we are running clinical validation, but also developing the at-home sample collection kit. So in the future, patients don't even need to go to a urologist clinic. They can just pee in a cup, ship it out, and so they handle the rest. So yeah, this is our team. I was very fortunate to meet with my co-founder through Kevinoy class. I met Dr. Igor Sokolov, who is the inventor of this technology, went on to recruit 2 more scientists. In total, I think we have about 60 years of atomic force microscopy experience, and then a fantastic advisory board who are diagnostic veterans and really helping us to think through the pathway where we can bring this product to the market. So thanks so much for having us, and happy to take any questions.
Any questions?
Yeah, first, so I'm assuming these are circulating tumor cells.
This is just the epithelial cell that lining the bladder.
And you're taking it from the urine.
From the urine, just like a.
These are circulating tumor cells which are shedding
Mhm.
primary tumor, is that right?
So kind of like a urine cytology, right? So we centrifuge out the cell from a urine sample. These are the epithelial cells that they come from the tumor, or maybe other cells that lining the bladder. And there is a lot of research that show that bladder cancer is a field defect kind of cancer, meaning that when there is tumor, the whole bladder kind of spoil, so that we see the signature of cancer in those cell surface. When we get those cells and then we touch them, it's actually feel like it's not normal. It's not looking, it doesn't feel like from someone without a tumor in the bladder.
Yeah, the reason I'm asking is that when you compare your data, what very impressive 94% to the 74%, I'm assuming the 74% is because they're doing that invasive procedure, which obviously I would want to happen to me.
Yeah,
Obviously,
me too.
So I'm just trying to figure out, it looks like it's two very different approaches.
That's right. One is a urine test and the other is a procedure. And
One's looking at primary tumors, one is looking at essentially circulating tumors.
Yeah, it's because the cystoscopy, that is a visual inspection, right? So you put a camera down the bladder and you see which, tumor that could be in the bladder. So you could miss cancer because there are smaller tumor that you may miss as a urologist, or there are some just inflammatory, but you thought that is cancer. So there are opportunity for a urine-based test like it to come and then be able to show that it actually not cancer or it actually cancer, and help urologists to manage the patient there. But yeah, it's kind of comparing apple and orange because we are urine-based test and they are procedure.
It looks like there are a couple of other tests that have a non-invasive approach. That it's great that you have existing codes to use. That's wonderful. How does your performance compare to those tests, and what would give a lab or a physician a reason to use atomic force microscopy versus a molecular method?
Yeah, I asked that exact question to my urologist. So the first time I met them, like, do you have anything else besides cystoscopy? Like, really, you just sticking a camera? And I asked that question many times. The answer is no, Jean, I told you there's nothing except cystoscopy. We can use Eurovision FISH or CX bladder, the two tests that I show you. They give some genetic mutation which correlate with high risk of having a recurrent, but it doesn't tell you exactly if the patient had recurrent or not. It's tell you the risk, right? Because we are genetic testing, and there are some protein marker that urologists could use just to help them to see if there is any higher risk of the patient, so they look more carefully under cystoscopy. But in terms of the accuracy, the best test that we found on the market has around 80% accuracy through like 10 years of clinical review. That's the best that urologists could get. But that 80% is actually just good for high-grade bladder cancer, which is only 30% of the total bladder cancer case. So there is a unmet need with a more like a higher accuracy test and also non-invasive approach to managing patient. Yeah. I don't know who's raising their hand, but okay, we'll go with you.
Thank you for presenting. It's very cool. Are there other cancers for which you would anticipate using your technology, or that you're considering working on?
Yeah, we actually started with cell line from cervical cancer, and realized that obviously we can do that. We can detect with 99%, but pap smear or other tests already on the market. So there wasn't like a clinical urgent need for that type of cancer, even though we have really good data. So like I said, because this approach can work with bodily fluid, so urine or saliva or other kind of bodily fluid where we can get cell would be a good targeting for our test, but we would need to do more market research to see what is the next one for us. Yeah, and
so I understand that you're using the AFM method, but I guess I still don't understand the role of AI. Because I guess the question here is go, no go. So cancer, no cancer. So what's exactly AI doing here? And the second question is, how do you determine the accuracy of your AI method in here, like with other?
With biopsy.
Yeah.
Yeah, so there's two parts, right? What is the role of AI? So if any one of you working with AFM in the room, you can raise your hand. Okay, perfect. So you know that we're generating a topography map, physical property, right? But you cannot use that imaging with your eyes to see like, oh, these are, roughness survey, or it look like cancer. It's impossible to look with the eyes. But when you convert it into survey parameter, which is quantitative number, you have quantitative number to train the machine learnings on different channels of the AFM, not just adhesion. You can add disconnection length, you can add high different survey parameter. That machine learning is really good at learning, okay, if you give me these survey parameter that you convert from AFM image, I can learn, we use supervised machine learning, I can learn and then can tell it's a binary score, yes and no, that this is cell coming from someone that have cancer or cell coming from someone without active cancer. And it helps us to process a big data set that is impossible to do with statistic or with human eye or to other methods.
Okay, and also, are you using AI to find the trend or just like finding the pattern?
Just a pattern.
Pattern.
So we compare the our score with biopsy, which is the final truth of having cancer or not, and then we show that on the testing set, after we train the machine algorithm, you would scan someone's cell, right, and it would give you a score of yes and no, and you compare it with biopsy, and it's 94% accuracy. Oh, yeah, sure. Yeah, go ahead.
I think on the last question in the room, a lot of the question has been asked already, so I have one question left. Regarding your approach looking at the morphology of the tumor, so with the atomic the functional microscope approach, what are you looking for on the surface? Filtration methods will look at cellular level and then determine the biomarkers. I think there is a reason looking at cellular level giving accuracy in cancer detection diagnostic. This is great, it's also a large problem, but there's also false positive detections that are right now happening, giving you other issues as well. So I just wanted to see, would you think your approach will be a supportive diagnostic method, or do you think that in the future this will be a replacement for diagnostic, molecular level diagnostic for solid tumor?
So if I understand your
two questions. The first part is like, what are you looking for on a morphological level in terms of what are you looking for cancer diagnostic? Second level, would you think that this will be a treatment that will be supported with the diagnostic in the current market, or do you think that it will replace the current diagnostic treatment?
Yeah. I think I understand both. Okay, let me try. I think for the first one, we're not only looking at the morphological level of the cell, because you can do that with optical microscope, right? You can see nucleic and other thing under optical microscope. This is more like a 3D level. You see the whole surface of the cell, the property, like imagine if you're touching something and you feel its softness, and it's a number, it's a value for the softness. So it's not just looking at the cell morphologically, but you can understand its physical property through this AFM mechanism. It's very novel. I think we're the first one that use AFM for clinical diagnostic. And the reason for that is because it has been known as a research tool, but now automation and other kind of advancement in the AFM make it so easy that anyone can operate this machine, and it makes sense to use it for clinical diagnosis because you have so much information per cell, way more than an optical microscope could have. And second part of the question, I think molecular diagnostic, they keep a different kind of information, right? There are some protein marker that pathologists or urologists still using to detect cancer. There are other liquid biopsy that have ctDNA or other kind of way to try to understand the cancer. For us, I think we can be complement to the modality, but obviously will be replacing some of the tests that do not have that accuracy and do not have that clinical utility for clinician. So like any disruptive innovation will replace some of the tests on the market and I guess be a new standard of care for managing patient with cancer.
Thank you. Thank you. Appreciate them. The wonderful questions. Thank you. Wow, a lot of good questions. So I think we're going to take a little brief pause from the showcases and want to just touch upon something that we wanted to highlight and recognize there's been a lot of changes in the past couple of years. And what it really enabled us is to put a spotlight on life sciences and what technology and healthcare can do when combined together. And so we wanted to take the opportunity to invite our distinguished guests here and industry experts to a retro, a post-COVID retro on reimagining the life science cycle of funders and builders that Matt Carlin would lead. And Matt Carlin is a partner at Morrison Forrester and the co-chair of the firm's life science transactions and licensing practice. He has had 25 years of experience in working with companies in the healthcare, pharmaceutical, medical device, and technology industries on a wide range of commercial life sciences and technology transactions. He's consistently ranked as one of the U.S. best lawyers and one of the most respected licensing attorneys in the country, and has been a key supporter of the Boston Life Sciences Showcase and with our partnership with Morrison Forrester. So really, thank you.
So can the panelists join us? Skip and me. Come over here.
Good to meet you.
Michael. Great. This is a little not fireside chatty, but we're gonna pretend. All right, great. So we have a big task in front of us. So the title of the fireside chat, Missing the Fireside, is reimagining the life science industry in a post-COVID world. I've got 7 extraordinary guests here today. We have that task to accomplish, and my dear colleague Maya Zamaris has given us 25 minutes to do this. So we're going to try to keep it to 25 minutes. Not sure how much we're going to get through. We'll try to leave some time for questions. Hopefully that works. But I'll introduce this by saying, well, the life sciences industry, pharma, biotech, med device, digital health continue to experience significant growth. The headwinds are strong, from global political unrest to economic uncertainty to the seemingly endless pandemic and the impact it has had on supply chains, work habits, and the ability to attract and retain talent. While the scientific and medical communities work with the government to continue to find effective measures to fight COVID, the impacts of the pandemic will be felt for years to come. As we emerge in a post-COVID world, how have we changed? We will spend the next few minutes asking industry leaders, my distinguished guests. I was about to say extinguished guests. That would pass it to. Hopefully we can keep you engaged and active for 25 minutes. We'll spend the next few minutes asking our distinguished guests to reflect on the ways life science companies have pivoted and adapted to face new challenges and ask what's next. So I'll ask each of you to very briefly, if you could, introduce yourselves, and then ask you from your vantage point as investors or lenders, entrepreneurs, advisors to emerging growth companies, let's think about the lessons and takeaways. And if you would, share your perspective for a few minutes on the impact the pandemic had on your company, or how you viewed investments, or the advice that was provided to companies, and 1 or 2 key lessons you have learned or observations you have made during or coming out of the pandemic with one key thought in mind. Where do we go from here? All right, so who's going to get the dubious honor? So unfortunately, Skip, you're first on my list.
Can you repeat that?
We don't have time to repeat.
I know we don't. All right, I'll be brief. So Skip Kelly, I'm with JP Morgan. I work with the venture community across life science and healthcare. I've been with JP Morgan a couple of years now, and really what we're doing is trying to support venture investments, portfolio companies as they need, whether it's general banking, venture debt, which I can speak about, But really what we're trying to do is work with companies at the seed Series A level. And I think that's a change for what I think folks think of as JP Morgan. Think of us a little later stage, working with our investment bank, but really we want to grow with all of the entrepreneurs, really early on, and then support you as you kind of grow and need more sophisticated needs. What I would say, the thing that I learned the most during the pandemic might be a little elementary, but I really focus on relationships with investors, relationships with entrepreneurs. And it's really easy to maintain relationships in the Zoom world or, connecting for 30 minutes, but it's really hard to build those relationships. And I think that's the biggest thing, whether we're not making investments, but we are, lending venture debt into businesses. There's nothing that can substitute sitting with the founder and just hearing the story, hearing the vision as to why they're excited about it. And that was really hard. I think there's utility in lining up 20 Zoom calls for 30 minutes each, and you can have a full day, but you really don't get to know the entrepreneur, the investor, why they're excited about it, and really what their story is as to why they started their business. And so I think that's one of the biggest things that I learned kind of through the pandemic. Is that good? Do you want me to keep going? Should we?
Fantastic.
Yeah.
Vineet.
Oh, I'm next. So, do I need this?
Your choice.
I don't think I need this.
I was told to offer it.
So, as a background, 25 years as an entrepreneur, tech entrepreneur. Companies that I built were acquired by Boeing, Motorola, Qualcomm, Mitel, so aerospace, telecom. And then 15 years ago, switched to being what I say is a grandparent, helping other entrepreneurs rather than being a parent. And that has taken two directions. One is teaching at Boston University. So I wasn't joking when I say I have a class, and two of these projects, one's in my class this semester, and one had been a few semesters back, and investing. And I'll say two things about the COVID. One personal, I'll never go back to a gym. When we had the great outdoors, we learned how great it is to just stay fit outdoors. And then I would actually say that as an investor, we found that we were much more productive over Zoom. And we did many deals having never met the entrepreneurs. We actually invested in them. And a small group of 3 or 4 of us did 63 deals last year. It's better for the entrepreneurs. They don't have to waste all this half a day coming downtown, paying $40 for parking, just to be told no, which we do 9.5 times out of 10. So it's quick.
What was lost in that process?
I don't think a lot was lost. We're back to sort of in person, and I'm finding that I'm less productive now. I spent 45 minutes coming from Kendall Square to here.
Yeah, we're so happy to have you. And Jaswinder, your thoughts?
Yeah, so just by way of background, my background is in science. I've worked in biotech, I've worked in pharma, and my first company I started in 2007. I basically lost my job at Biogen. They'd restructured, and I went to my wife and said, "You know what I'm going to do? I'm going to start my own company." And she looked at me and thought, "Are you sure?" And I was sure, and I was convinced that there was a better way of creating drugs, and that I could do it. I went out to investors, was able to convince them. Basically, we were able to raise a $20 million Series A from that first conversation I had with my wife. 12 months later, we raised $20 million and started a company that was 5 years later acquired with a platform that I think has revolutionized the treatment of cancer, leukemia, chronic lymphocytic leukemia, and lung cancer. So for me, I'm a big firm believer in entrepreneurship. I believe you can actually make a difference. And I say, in terms of looking at COVID and the impact that it's had, I'm actually a little bit sad and disappointed. It was a very disorienting experience to go through. I'm sure, like everybody else, who've had personal losses during it. I did as well. And for me, prior to COVID happening, I organized a Gordon conference on the problem of drug resistance and the fact that there were these looming threats, infectious agents that we weren't taking seriously. And I remember trying to raise money for that meeting. Went to all the pharma companies, went to the Gates Foundation, and we had like the world leading experts on infectious agents around the table. Couldn't raise any money. The person who made the biggest contribution to that meeting is talking to you right now. And I remember at the end of that meeting, people who were involved in the area of infectious diseases said, we're getting out. There is no future being involved in infectious diseases. And then COVID hit the following year. We wrote an opinion piece in Science Translational, basically warning people that there was an issue. And for me, I look at it now, and I'll be honest with you, I think people move on very quickly. I think the financial markets moved on very, very quickly. And we talk about the impact in some respects. It wasn't like a long lasting impact. I mean, you probably did a lot of work subsequent to the pandemic. So I think for me, I don't think it's gone away. I think that and part of the problem is the business model. We talk about entrepreneurship here. There is no business model for infectious agents. And when people look at Moderna's business model originally was not infectious agents. So to some extent, they were able, well done to them, take advantage of a one-in-a-century event. And the problem is there are other one-in-a-century events, I think, looming, and we're not taking them seriously. So that's my concern.
Michael.
All right. To me, the most interesting observation about the pandemic is that it's probably the most comprehensive natural experiment that we'll see on success and failure in biotech. Everybody wanted to develop a vaccine or a therapeutic. Our own company set about doing it, and before that we were doing CAR T cells for cancer, okay? And so we talk a lot about the few great successes, for example, with vaccines, but there was a vastly higher number of failures. And so I think it confirms what we already knew, that this is a very risky business. And even when you take the smartest people around, it takes a lot of luck, right? I think that's what I'll say about that.
Good insight. And Kate, you come from a little bit of a different perspective, from a larger company, and curious to know your answer to the question, your thoughts, but also coming from a significantly larger company perspective, is there a silver lining to your business? What has changed for the better, and what perhaps has not changed for the better?
Yeah, great question, Matt. So my journey to MilliporeSigma began at the bench, like so many people in this room. I have a PhD in immunology, spent 10 years in consulting, working for a lot of innovators, a lot of investors in this space, and love that enthusiasm and the passion project that you all have. I had the pleasure of joining MilliporeSigma in March of 2020. So great time to get to know a lot of colleagues on Zoom.
Was it before? Were you ever in the office?
I was in the office for 2 weeks. Yes. But what I saw from that experience so quickly after I joined, getting used to the bigger company mentality, having come from, a small 150-person firm, working with a lot of small venture-backed biotechs and life science startups, was actually focus. And a lot of large companies, because of the breadth of ground we need to cover, inherently lack that focus that drives so many of you here in this room, because it's your single vision. It's the first indication that you're pursuing. It's the first-in-class technology. And unfortunately, for better or worse, large companies have the resources to pursue so much. But it's like pulling teeth to actually get people to unite and decide, like, this is the one thing that's our priority. And so the pandemic gave us a couple of things that were definitely our priority: supplying our customers, making vaccines, therapies, and tests to counteract COVID, That was a huge pressure on our operations as well as many others in the life science space. The scale of demand was just not what we had been projecting and planning for. And frankly, we were caught a little bit off guard. But emerging from that, I think it's also given us that focus on just making sure we have a resilient operating network, because we felt the pain of supply chain issues. And I think that message of focus really, as I look to like what's over the next 12 months, 18, 24 months, I think it matters a lot for smaller companies too, because R&D and venture-backed funding, you know, we've seen some contraction so far. I know that's not felt across the board, but with so many macro uncertainties and higher interest rates, it's something that companies are going to need to tighten their belt a little bit and choose among these three assets, what are we going to prioritize? So I think focusing on impact is a good positive lesson for all of us through the pandemic.
And do you fear, from the earlier point, that we have short-term memories and that we don't learn from history? Is that something that you talk about internally at all, that you say, you know, we need to be resilient, we need to be prepared, we need to learn from our lack of preparation before COVID? But the point was made earlier that have we just, or is there a point at which we're just going to forget that it happened?
And there's a point at which quarterly earnings at a large public company that's the drumbeat that you have to march to. But it doesn't stop us from committing to some, you know, more of a shift to an in-region, for-region production footprint that we're definitely committed to.
I don't think we're going to forget.
I don't think we're going to forget.
We're all scarred.
I mean, to an extent, we didn't forget the great flu epidemic of the early 1900s. So, 100 million dead. Here was 10 to 15, depending on how you count it. If those vaccines hadn't come within a year, it would have been closer to 100 million. We're not going to forget that. And I think the other thing that people are not beginning to realize is that there's systemic risks in the planet. Climate change is connected to pandemic, is connected to geopolitical, problems and so on and so forth. I don't think we're going to forget that.
Yeah, I think coming back to what I said earlier, I think the proof of whether we forget or not is whether we solve the problem of how do you develop drugs for infectious diseases, which are not large markets, for which you can't easily predict when it's going to be a problem, that specific agent. There just isn't a business model. Companies abandoned in the 80s and 90s their infectious disease research. Everybody suddenly got into cancer because that's where the money was. And the problem is that, the big revolution in infectious diseases was also the development of antivirals. Gilead was a leading force in the development of antivirals, and yet they're now more like a cancer company, right? I think to some extent there aren't that many quite huge players actually developing drugs for the next problem, viruses or bacteria. That's my concern.
And just along that same line, and I want to get to Clark and Murray as well. Michael, you said that there were winners and there were a few winners and there were a ton of losers. What was the tipping point between being a winner or being a loser?
I think that it was arbitrary. I think there was a lot of luck. Now, of course, we look retrospectively and we see mRNA technology is terrific for this purpose, but we didn't know that prospectively. And I think the point is everyone in this room is a risk taker, right? And we're being asked to take on enormous risks.
So that's with respect to the vaccines. What about with respect to therapies, with respect to testing, at-home testing? Any tipping point there between the companies that were able to capitalize on the moment? That's probably the wrong phrase to use in that, but able to take advantage, that's probably also the wrong phrase. Able to win versus those that tried and just simply couldn't make it happen.
The diagnostics were trivial. The technology we already understood. The therapeutics, again, the technology was understood, and I think that was just good old-fashioned drug development. I don't know if anyone else on the panel could have spotted the winners and losers ahead of time.
Yeah, interesting. Clark, welcome.
Yes, sir.
Yeah, let's get you in the fold here. Your thoughts?
So by way of introduction, Clark Dees at Teneo Capital, which is a boutique investment bank within a broader consulting firm. I lead our healthcare practice where we focus a lot on emerging growth companies in life sciences, both on the product side and the services side. I think it's already been touched on in from various perspectives, but I think the single biggest takeaway for me and for us about the pandemic within this space, within this sector, was just the resiliency, right? I mean, from a financial services or markets perspective, the fact that there was barely a pause in fundraising, right, in sort of early Q2 of 2020, and then the greatest record-breaking pace of fundraising across public and private, various different methods or instruments that there's ever been, right? And obviously that was in part attributable to funding of potential vaccines or therapeutics, less diagnostics, as Michael says, but by no means exclusively. It was broad, right? And it wasn't even just infectious disease. I share your concern that it actually didn't spread that far beyond COVID, but it spread across the entire sector, right? At a time when you would have expected life sciences to be impacted by some of the other, just the same way as some of the other business models in the healthcare space, right? I mean, clearly not every drug is required today, and how do you get new prescriptions, and how do you get on cycle with new drugs if it requires a doctor's office visit and it's a medical benefit if doctor's offices aren't open, and yet the funding reached historic proportions. And so to get to the last part of your question, and I'll stop being long-winded, in terms of what does this tell us in terms of look forward, it probably tells us we can be cautiously optimistic as we look over the lip at the various macro concerns, recession, geopolitical risk, and the like, and not be as negative as some are in the day-to-day, because if you take a historical perspective, no, this doesn't look like a fundraising environment that we had in the first half of 2021, but nothing does, right? Relative to the last 5 or 10 years, this is still a pretty healthy fundraising environment for private companies. It's a much more targeted and cautious one, but there's still pretty healthy flows out there. So I guess I'm not typically the cheerleader in the room. I'm more of a Sergeant Friday person, but maybe I'll play that role today.
I wasn't expecting this panel to be so positive. But that's a great segue to Murray, who I know the best of everybody on the panel because he's one of my partners here at MoFo and leads our emerging companies and venture capital practice globally. And Murray advises hundreds of companies in this space, really across the global economy. And so, Murray, I know you have a wealth of knowledge, and I'm certain that the entrepreneurs in the room and investors in the room would love to hear your perspectives.
Well, let me say this. It's really hard to go seventh. It's hard to go seventh to try and build on all the really smart things that everybody else said and additive to it, because there were a whole bunch of things I was going to say if I was going second and fourth, and I was saying Carlin's going to make me go last because that's the polite thing to do. Let me suggest a few things if I can. First of all, my wife a long time ago told me to keep it simple, stupid, and so I'm going to try that, and I'll offer when we break out to talk individually with anybody who wants to visit. I think the MacArthur Foundation should come to this room and just dispense all its money to the room, because literally everybody before me who has spoken has said such smart things that I don't begin to understand, but it's quite clear to me that there are people in this room solving problems that really will make the world a better place. And I thank all of you for contributing your lives and efforts to do that. I spend a lot of my time with tech entrepreneurs who are coming up with the next clever app or not. Who cares, right, compared to the sorts of problems that you're all working on and solving. Secondly, for those who were listening carefully an hour ago when Kevin kicked us off, anything and everything I want to say, he encapsulated in a sentence or two that went by really, really fast. And I'm going to tease a few points out that resonate with me and don't have anything to do with science, but somebody's got to be the village idiot up here, and it's me. For those of you who are raising capital, first time or 10th time, I would encourage you to have a question in your mind that Kevin posed, and I'm going to state it slightly differently, which is, why will somebody join your company as the 100th employee? And for those of you who've never thought about that question, which sounds deceptively simple, which is why the professor put it to you, and some of you got it and some of you didn't, the person who joins your business as the 100th employee will make no more money than she will make at the largest and most successful company in the world. The value of her equity will be nothing compared to those of you who are founding businesses. You have to do something super, super special in order to attract people to join your company at that stage and to stay with it. We face that problem at MoFo every day of the week. Matt, Mike, and everybody else who's in here knows that challenge exists. And while the science that all of you described is great, and again, I don't begin to understand any of it, I promise. That's not false modesty. I assure you I don't understand it. I do understand that you will all need people to make your business work. And how you figure out how to be kind, and how we all learned, see Matt, I'm bringing it home, how we all learned in the pandemic how to be kinder are lessons that I encourage all of us to think about every day and figure out how to apply. I better stop there.
Thank you, Marty. So I know we're butting up against time, but maybe one or two questions from the audience? Anyone for our panel? No one? We have a question.
So no one for Beneath. So you were talking about deal flow over Zoom during the pandemic. I'm curious as to how that has affected the geography of deal flow. Whereas, has it been centralized into certain hubs, or have you been able to go in between, or I guess to like fly over cities?
Well, first of all, we're only focused in Massachusetts.
Okay.
I would say that we actively try to get deals outside of Boston and Cambridge. I don't think pandemic or Zoom helped that.
Okay.
What has helped, though, are the investments the state made in UMass Amherst, for example. They built a life sciences center out there 8 years ago. And now it takes that long before things sort of gestate and start. Now there's like interesting spin-outs coming from there. But honestly, I thought we would see more deal flow from other parts of the state, but I don't think it actually made a difference.
We've done maybe a dozen deals with people we've never met in person, and we did a deal in Kansas. We wouldn't have done otherwise.
That makes sense. Yeah. Thank you.
Another question back, yeah.
For anyone on the panel, I wanted to know what to you or what you look for in investment for the concept.
I mean, so most investors look at three things, and they emphasize one of the three according to their own philosophy. You know, the first thing is the quality of the science or the technology. The second is its market opportunity. And the third is the team. And people emphasize, as I said, different parts of it. What do you emphasize?
Oh boy, all three.
I don't emphasize all three. I emphasize people. But you know, because I was an entrepreneur for so long, I just have a natural affinity for entrepreneurs that I think have to make it. Like some of the in this room.
Anyone else want to take a stab? Kate, what do you, when you do collaborations and biz dev on that front, is there something in particular you look for in your collaborating partners?
Yeah, I think wearing my Millipore Sigma hat, we come from a very different side than the venture investors. And so we're looking at the caliber of the science and how it fits with what we're doing internally, because it needs to align with the gap there. And I'd say that's probably true of a lot of other larger pharma companies as well.
Yeah. Anyone else want to chime in? All right, a question. I think we're okay. I can see Laura is like eager to. Do we have one more question? One more question.
So it's a broad question maybe for everyone. If today you were starting a company or investing in a company, what area would you go in? There's lots of buzzwords out there.
Let's go down that. Let's go down that. Let's start at the end and see what the answers are.
Well, I think that there is this need for infectious diseases. But I'd probably go into autoimmune diseases. That's where my emphasis would be. Yeah.
That's it. Want to bring it down?
I would be safe. I would do oncology.
I wasn't necessarily thinking according to therapeutic area. I was thinking more in terms of modality or platform. I think Tailwind's certainly the greatest behind precision medicine broadly and therapy.
I'll depart from therapeutics entirely and say life science is a really broad market and climate resilience is a really big challenge. So biological approaches to help improve our environment.
She just took mine.
Maybe we can team up.
That's exactly what I was going to say. I'm focused on clean energy, and for this audience, I would say anything that's biology driven.
Nucleic acid delivery.
Oh, very specific.
I'll pass, man.
Okay. All right. Thank you all. That was a great chat.
Wow, okay, so some incredible insights from the panelists, and we're not done yet, so everyone else take a seat. We have 2 more showcases to get through. The other room to break away. It's fine, we'll continue. Right? Okay. So without further ado, I don't think they can hear me. Everyone, if you don't mind taking a seat, we have 2 more showcases. Hate to be the one to do that. It's not fun being up here, dude. So we have 2 more showcases for this evening, and without further ado, I'll go ahead and introduce Kwame Su from Hopewell Therapeutics to give his presentation.
Thank you. So I need to say goodbye. I received some promotional consideration. So first, I would like to thank Kevin for the connection and say goodbye for the invitation. So you give me the opportunity to introduce our company, Hopewell Therapeutics. So we're actually a delivery company, in particular focus on the nucleic acid. So that's our slogan. We deliver hope and wellness to our patients. So a little bit background about myself and also the company and how did I get to here. So I got my PhD at Harvard in chemistry, and I did my postdoc with Professor Bob Langer at MIT working on the delivery. So during my training in 2007 to 2010, so I worked on the LNP delivery for the protein and also mRNA delivery. At the end of 2009 and also the early 2010, that's where Moderna started. And I started my own lab in 2010, almost a similar time at with Moderna, started their company.
So I started my lab at Tufts in the BME department as an assistant professor. So, but it's totally that Moderna is a multi-billion-dollar company, and I'm still a professor there. So based on my postdoc training, and I know that I have the same similar vision or passion to Michael about the delivery. So that's one of the important things to actually achieve the therapeutic potential of various of the nucleic acid. So in 2013, so my group mainly focused on developing the novel LNP delivery system. So we filed the first IP in 2013 and about the disulfide library of the disulfide lipid-based delivery system. And based on this system, and we later we have expanded our library, and also we have used this for various of the applications, including gene editing in liver, lung, spleen, T cells, lymph node, and also the brain target delivery. So, and back in 2018, like March, so we received the angel fund, and we incorporated this company called Hope Therapeutics, and we licensed the technology from Tufts. And not until last year, September, we successfully raised like 8 million seed round of financing, and I took a year leave of absence to run this company and to build this company. So after 3 months, we received 7.2 million, the seed plus round. So with the total 15.2 millions, led us to actually build a 25 people team, 10,000 square feet operation in Woburn, Massachusetts. So that's sort of the background about the company and also myself. So what we do, as we all have hopefully received the COVID vaccine, so that's the mRNA and the lipid. So the lipid there is to deliver the mRNA into the cells to make the antigen, which is spike protein. So the lipid is one of the very important things to achieve the therapeutic potential of the mRNA. As you may also know that in the lipid area that Alnylam sued Moderna, Pfizer for the lipid chemistry, all those things, right? So we believe that the Hope has a very unique chemistry, which is very different than what Moderna, Pfizer are using, because we have a peptide like disulfide bond, selenide on those lipids. So the composition is completely different than what they are using. So we have using our chemistry, we have a very clean IP, and in terms of those lipid composition, we have a library of those things. So we utilize the rational design and modular synthesis to generate a library of those molecules and integrate the high throughput screening to achieve the specific organ and the cell selective delivery. And this is sort of the platform that Hopewell is building. So we call it Atlas, like a platform. So Atlas is collection of the maps, the route from here to Hope there. And using, if we actually zoom into like a cell lab or the human body in the organ and the cell lab, so how do you deliver the cargo into specific cell and specific organs? So that is a map. So whether the cargo is a messenger RNA or gene writer or sRNA or circular RNA or other biological cargoes, you all need a delivery to deliver those things into the targeted cell for the therapeutics. And so we want to put those cargoes into the our LNP the library, and then conduct the library screening and identify the lipid that can achieve the specific organ and the cell delivery. So what so in the next, I show you some results that we already achieved, but I would say that we only achieved partially, but not completely. Our goal is to achieve the complete or the Atlas. So those are some results that we showed here using from the lipid from our library. So this is the luciferase. delivery. So for example, this lipid can exclusively get into the liver, and this one can get exclusively get into the lung, and this one can exclusively get into the spleen. So that's just by changing the lipid type in the formulation, and we can achieve this organ and the cell selectivity. We also validated, for example, the lung delivery in the mice and in larger animals like the rabbit. So it's pretty exclusively in the lung of the rabbit. And we are building some new delivery system that's even better than Moderna, and here the BioNTech's lipid. So for example, we can deliver the various of the cargoes into like brain. So in this, we also publish the results have an IP coverage. So we generate a new class of the lipid. So based on those neurotransmitter derived lipids. So we showed that we can deliver the small molecule antisense oligos and also gene editing proteins and so also the mRNA into the brain of mice. So in the nucleic acid delivery field, and especially if you use this modality to treat like metabolic readiness or some modality, repeated dose is very important. So we also conduct a head-to-head comparison with Pfizer's lipid and Moderna's lipid. So that's a lipid from our library, and you can see that when you inject multiple doses, the protein production, so they actually get constantly decreasing for some formulation. But one of our formulation, we find that the protein production are pretty constant. You see that with the Moderna's lipid and also the Pfizer lipid, there are also some decreasing. So this is sort of the summary about our lipid delivery system and compare with our competitors. So like the vaccine in this area, is the lowest hanging fruit because it requires small dose intramuscular injection. But if you want to use like the mRNA to treat the other diseases like the liver, lung, brain, kidney, or other like heart muscles, so the delivery is more need to be more sophisticated and advanced. So for the competitors, it's based on so like the vaccines, the Pfizer, they got into the FDA approval. So we also have done a head-to-head comparison with their lipid, their formulation. So we can do the same thing, but even better with better efficiency and efficacy. But in terms of the marketing, because they are giant. So as a small company, we will not compete with them. So we are started with delivery platform technology company, but we also we need some kind of cargo in order to put them together to make a therapeutic drug. Right? So currently we are working with we are actually developing our own cargo. So those are like internal wholly owned cargo, and we're also approached by many companies to build a partnership with our delivery system. So that's sort of what we are working. And finally, so this is our team, and some our CSO Katie is over there, and she has multiple years experience in drug discovery, development, and also the former VP in the gene editing company Editons. So that's our profile. Thank you.
Great presentation, and yeah, really excited to open it up for questions. There you go.
So very cool. Have you been looking at ways to tag these LNPs to target them to particular cell types or tissues? Yeah.
So we didn't tackle it, but that's certainly one of the strategy trick you always can play. So what we did is just change the active lipid, the identical one. So we find that depending on the lipid type, they can actually, even they go to the same organ like the lung. We have more than 10 lipid can go to the lung. and we find that the cell population again two are different.
Yeah. So Sheldon, it seems like one of the unique properties of your system is your ability to cross the blood-brain barrier, carrying therapeutics that may already pre-exist. Does that create a real differentiation for you?
Yeah, so that's one of the differentiation. Or we are able to, and the existing lipid nanoparticle cannot, right? So, that's potentially that we can use our stuff to treat, for example, neurologic disease. And you see, for this platform, you can do a lot of things like deliver small molecule, small nucleotide, or mRNA, or circular DNA or RNA. So it can, but from the business point of view, whether we should actually use the existing drug and then to pack them and to get into cross the BBB and to treat, for example, cancer. So technically we can do that, but from the business point of view, we need to discuss discuss whether that's the best one to actually convince the investors, say whether that's a top-notch or the best application for our technology.
Thank you.
Any other questions?
So you mentioned about strategic collaborations that your company are working on. Could you just disclose a little bit about who other companies and what other therapeutics that you guys are working along with your platform?
So I will not say the name. Okay, so I will say the company's type. So they are like a larger pharma or working on multiple pipelines or those companies. And they are also like star, but star like biotech companies. So they're working on a specific area in the gene manipulation or those things. So they need a delivery, right? And they are also like many like smaller companies. They have certain ideas, but they need a delivery. So those three parts of the companies are approaching us.
Perfect.
And do these, are you partnering largely around potential novel drugs, or are these new delivery mechanisms for existing drugs primarily that they're thinking about?
So actually for the mRNA field, the mRNA itself is not a drug if there is if it's not associated with a specific delivery system. So if, for example, the client or the partner has a cargo that's pretty proprietary for their company, could be like gene editing, let's say. And so they're working on the gene editors, and they need a delivery to cross or to reach to the certain organ and tissue, right? So we can help them to do that, or we can form a partner. Or if the company, really that it's an existing drug, they want us to help them to do that. It's more like, I think that probably because of like what Michael was so the next generation is in the nucleic acid area. So that's where the market need for the delivery is more than just like a reformulation of the existing drug.
Great. Yeah, and then I'll wrap up with the final question here. Just wanted to ask, what was, you guys have had a lot of great milestones so far. What was one of the ones that really, you felt most proud about, or, most excited to achieve?
Scientifically or the corporate building wise?
However you want to answer.
So, that when you are a professor, right? So you always write grant to do the medical proposals to propose, to get the grant to do the research, right? So when you run the company, I took a 1 year leave of absence. So it's sort of the resource-wise, it's more, and you can actually bring more well-trained people and with different expertise to put the company together. So for example, in our company, we put the chemistry, and also we have the formulation group, and we have the RNA biology group, we have the scale-up process development associated with QC, and we have people working on early drug discovery, like pipelines. Right? So with a company, I think that the resource, you can put all those things together. I cannot imagine in my research group we can have that many experts working those things. Right? So that's the most impressive or proud thing that I feel.
Amazing. Yeah, great job. Yeah, I'm really excited to wrap up with our last presentation of the night. Is a company that's already raised $95 million in the Boston area, Click Therapeutics.
Great. Is this on? Yes, okay, you guys are still awake. I'm what's keeping you from the bar or asleep or right or your bed or something like that. So Shaheen Lakan, full disclosure, Laura works for Click Therapeutics, your MC today, right? But you put last, so right, that says a lot. I'm also her boss, which is interesting. Okay, so I'm Boston-based. I'm a neurologist, pain management physician. I don't want to read you my CV, right? But I've done it in medical education. I actually still keep my professorship in neurosciences, neurology, and internal medicine from various institutions. Was in healthcare administration. Had this wild adventure with the DaVita Health, the largest kidney system. So their COO, David Klein, and I formed a company called Savita Health. And we didn't get sued. And then we did addiction medicine practices across the United States for the opioid epidemic, benzo epidemic, alcohol use disorder. I mean, for a moment in time, I actually sat on Medicaid, Virginia Medicaid P&T committee. So, the ones that make formulary decisions based on new treatments and diagnostics. I think it was $8 billion a year. But now I find myself to the subject of this talk, right? So Chief Medical Officer of Click Therapeutics. So who in this room has heard of digital therapeutics? If you could raise your hand. I'm loving this. Okay, great. So that's like maybe 70% of it, right? 70%. And let me just ask you, Mike, I'm gonna put you on the spot over here. What are digital therapeutics? They're certainly not nucleic acid delivery systems.
Well, I think they're computer implemented methods and software to provide a therapeutic benefit to a patient.
Yeah, my job's done. I don't it's over, fund me. No, that's exactly it. So the applications that sit on your smartphone, hey, this is my drug delivery system that just happens to be we have digital drugs and their applications that sit on your smartphone. And yeah, they exactly do that. They treat, manage, or prevent a disease or condition. That's the official diagnosis, or something called the Digital Therapeutic Alliance that standardized this nomenclature. That's what I do. Okay, let's move to the next slide. This is nice. Laura developed this. 3 phrases here. So software as medicine. Now, for software to truly be medicine and not just be a marketing term, it's not going to sit all the health and wellness applications that sit on your phone, right? Those are typically trackers or give you some healthcare facts. They might even call themselves CBT, but they're mainly psychoeducation that just translates some literature out there and puts them into some content there. Worse, they don't have clinical data, right? This is an evidence-based room that wants to see trials. And what I love is I've seen a lot of trials thus far, right? Data. I mean, some of it was preclinical, and hopefully they pan out in human-based studies, but that's exactly what we do. In order to be on the same footing and have a new category of medicine, then I'll talk to you why we need a new category of medicine shortly, but it's all the evidence package, multiple randomized controlled trials. You got to prove that you have a mechanism of action. You can't just say it targets everything. You got to have target engagement, and you got to have a dose, response relationship. Basically, what am I saying? The tenets of drug development and drug discovery and development. That's how we approach it. Now, second, I already said deliver directly to your phone, but there are many ways to deliver things on your phone, and actually some of them touch on health equity, right? You could develop for the latest version of iOS, and guess what? You're really going to the most affluent population that's possible, and actually you're probably perpetuating a lot of the disparity gaps. The digital divide is amplified and things of that nature. So we actually, take a good approach in doing that and support all platforms. In fact, just by requiring a constant internet connection, there are a lot of applications that require that in order to function. You're already eliminating a tremendous amount of the global population, and dare I say even the US population. Digital divide exists here, right? The Affordable Connectivity Plan and Obama phones that meant to mitigate some of that. So we entirely operate on edge. So you don't need to have a 24/7 interconnected device. Last but not least, I think this is important. This is not just me and my ego as a neurologist, but prescribed by your physician. There is something like when migraineurs come to me, for instance, I see all types of patients. You know, if I prescribe to them or recommended to them pharmaceutical therapies, so there's drugs, there's exercise and lifestyle modifications, there's physical therapies, there's injections and procedures. I happen to be an interventional pain doc too. Like, why are they following my recommendations? Why are you? We're all patients, including me. You know, why do I follow my doctor's orders? Well, there's something called the therapeutic alliance. In fact, this is probably the biggest driver of effect sizes in every study in humankind. And then we can also say placebo response, right? It's because, one, they have faith that I'm a fully board certified, licensed, and it happens to be this, the Harvard healthcare system has employed me, right? So my credentials are check out and they accept it. But two, this is even more important. We have a shared functional goal, right? Your goal is my goal. This is often a disconnect in clinical practice. And if you take a series of steps, you will make a series of steps towards achieving that goal, and it'll become achievable. Now, how do you encapsulate all of that you normally have, you know, either in office visit or in a telemedicine visit, and put into a smartphone? That's kind of the nut that I think that we cracked, essentially. Okay, there are three large business models of how you can see, or delivery models of how you can see digital therapeutics. The first is obvious, and it's already out there, standalone. So use a application that sits on your smartphone as a monotherapy. It itself will treat the disease. And there are a bunch out there that can do that. And the most prime example is insomnia. Open every guideline from every agency, sleep medicine, family practice, you name it, even anesthesiology as an insomnia guideline. They all recommend CBTI. That's cognitive behavioral therapy for insomnia, but it's just inaccessible, right? Because psychologists are actually treating serious mental illnesses and other type of things they don't want to be burdened with insomnia populations. That directly can be put into a smartphone and has been put into a smartphone. So my colleagues, there are, say, competitors like Pear Therapeutics have applications that are FDA cleared called, you know, Somryst that treats insomnia. Second is complementary. Can digital therapeutics actually complement your standard of care or background medications? You know, biopharmaceuticals, that makes a lot of sense, right? Adjunctive use of it. In fact, they can play off of each other. Now, the complementary pathway, the only difference between combination is that they were separately studied and that they were separately approved in the United States through the FDA, right? So the drug got its own clearance, the medical device, that's what digital therapeutics in the US and the FDA's eyes are. Software as a medical device got its clearance. And the holy grail here, I think this is what's essential and has to happen to advance this industry, is the combination play. Combine these two assets, digital and drug or biological assets together. Actually, do it in either, you know, the same filing, an NDA or BLA type of filing. And the digital complement can actually enhance efficacy and reduce, you know, safety and tolerability type of events, right? Or negative tolerability events that go along with it. And it has to be adherent. So these are not adherence-based devices. They have to enhance the efficacy, clinical efficacy, and buttress against some safety things. This is what I'm going to talk about in a second. So I'm laying out the framework here. This is our platform at Click Therapeutics. And yeah, you know, when the font is this small, you're not meant to read it, right? It's supposed to confuse you. No, absolutely not. This shows the applicability of what we're building. So this is how I designed. I come from biopharma. I was Sage Therapeutics before, Zogenix, Allergan, Neurocrine, you name it. I probably worked or consulted there. But what we've developed at Click Therapeutics is what I call this digital mechanism of action library. This is the equivalent of active pharmaceutical ingredients. Some of these are very translatable. They've already been done there in academics. And what there's a literature pool, we translated it into a smartphone application module. Some of these are really proprietary and experimental. In fact, right here we have Dr. Lutz, who is at Biogen now. Unfortunately, she left my organization and my practice. Hey, but that's flattering. She went to Biogen, right? But co-invented many proprietary digital mechanisms of actions. Now, how you deliver them, these are what I call the digital delivery tools or digital tools that go, right? How do you get them into the compartment of interest, right? So they can have effect. And then what I have over here is Clicometrics is our data platform. How do you apply this combination of a digital MOA library and digital tools into different indications? And you'll see I am so hard pressed to find a disease that does not have a biobehavioral component to it. And therefore I can intervene on it. And I'll show you our pipeline shortly, but we started in addiction medicine. I'll spare you the story, but our founder had a serious addiction to the most addictive substance in the world, right? Nicotine, tobacco smoking, right? No, truly. And said, hey, we're all addicted to our phone. In fact, more often than not, if you wake up, you interact with your phone before your bedfellow. Think about it for yourself. So why not take that and use it in a harness for clinical outcomes? So he exactly did that with our first product, which is a smoking cessation application called Clicatine. We have since, far exceeded or evolved from that into psychiatric disorders like major depressive disorder and schizophrenia, into neurological indications. Migraine is our next lead asset. But you name it, you have it, including inflammatory disorders, atopic dermatitis. I would venture that's really more of a itch matrix, right? Neuromatrix that we could intervene on than a primary immunological disorder. But that's me, heavily biased. I see there's questions brewing. Good. Okay. Oh, this is good. Formatting issues for a tech company. Exciting. This is also meant to confuse you, but it's basically I was telling you what the pipeline is. But you know what actually came out was what I want to focus on. This is our discovery pipeline on the bottom. So what we have coined and what we are validating are something we call dynamos. Has anyone heard of dynamos before? There's probably two hands, right? Dr. Lutz, we co-invented this. Okay. Yes, we patented this exactly. So these are digital neuroactivation and neuromodulation. So digital neuroactivation and modulation, dynamo. I'm going to say a few times so it has some long-term potentiation, right, in your brain. But no, truly, when you think about diseases and you think about largely CNS diseases that have been treated based on the neurotransmitter paradigm, right? Upregulate or downregulate. And then, why do you have so many side effects and sedation? Well, that's how I'm treating depression, right? With something that actually suppresses a system for arousal and valence, right? No surprise to me as a neuroscientist. So we are developing targeted neurointerventions on circuits. And when I talk about circuits, I'm going to show you exactly what. So in depression, a major depressive disorder, why do depressionists see the world in such a negative bias and negative lens? I'm going to be very simplistic and reductionist here, but I'm going to argue that their amygdala, the emotional processing center, is hyperactive. We all get that, right? If something frightens us or something is painful and kind of activates this response, normally what do? Put the brakes on it. Our dorsolateral prefrontal cortex, the working memory over there, says, hey, that's not good for my behavior. There's other things that are more important than catastrophizing over, pain or sorrow or grief, and puts the brakes on it. But unfortunately, you have a hyperactive amygdala in depressioners, and you have a hypo, so not fully active, dorsolateral prefrontal cortex. What was cleverly designed was simultaneous activation of both of these nodes on the circuit actually builds neuroplasticity there and reestablish the circuit. So in summary, I know it's a lot of technical and scientific jargon here. Faulty circuits are responsible for CNS diseases. We have targeted interventions that fix faulty circuits. And the third thing is we put our money where our mouth is and validate that with neurophysiological studies, fMRI, right? And we have exactly that. So the data is published in Nature, right? Digital medicine and chronic stress and various other disorders in MDD. In fact, this is the product that we've partnered with Otsuka Pharmaceuticals, the multinational company that has almost created the market for incomplete responder depressions with Rexulti and Abilify. And our asset sits exactly on their pipeline like their drug assets. That's validating for prescription digital therapeutics. It's finishing a phase 3 trial, right? And by end of year, essentially. Great. These are our 2 models that we work with. Partnerships and collaborations. Otsuka is a perfect one. Boehringer Ingelheim is our next, our second one, we'll say. These are trailblazing. These are 9-figure type of deals with milestone payments, development costs. I have an entire in-house team of discovery, translational, clinical operations, clinical development, medical affairs, biostatistics, and the tech teams, which incorporates product, design, engineering, data sciences, right? We contract almost nothing out. And this is important. And you know, my CTO, Han Chu, who comes from Amex and Shutterstock and these more tech companies, I love like the perspective he brings because it's not a pharma background. You know, he said what we do well in our corporate retreat last week in Miami. He said, we speak pharma. And that's powerful because I see a fundamental language disconnect when a tech company is trying to actually develop therapeutics or partner with a pharma. They might strike a deal, but do they execute on it, right? And do they expand relationships? Do they go into a pivotal trial together? I haven't seen it here. This is what we have over here. And Boehringer Ingelheim, we have multiple products. So that's one, partnerships and collaborations and our in-house development approach too. Hey, now that we have learned through partnerships and gotten funding from a good amount of non-diluted funding from partnerships, we've actually built this machinery to do this in-house. Migrate and all of those dynamos are in the works. That's going on there. Okay. I am backed by, this is our management team, a pretty stellar team. I already mentioned Han Chu, their CTO, and his backgrounds. Randall Stanicky, many of you probably know him, particularly the JP Morgan side over here. 25 years as a sell-side analyst. He could have gone to any company, right? So he kind of chose us and said, hey, I've been creating white papers and opinions and other, press pieces on this industry. Let me actually join it and pioneer these efforts. Rich D'Annunzio was at Novo Nordisk, leading their endocrinology, right, insulin franchise, which is probably the largest therapeutic area that they have, and jumped on board because of the mission that we have here. Now, what's not represented is 140 FTEs, including Laura, right? That's part of it. And then our board, which includes the likes of Lee Shapiro, who had a fantastic exit with Livongo and Teladoc, just brilliant guy, great connections. And then Muzz Mansari from F-Prime, right, Capital. Hikma Ventures, Sanofi Ventures were early funders, Magellan Health and the likes. Cool. And that does it. And of course, I have to leave you with disclaimers. Exactly. Thank you. Okay, I'll take any questions. Oh, yes, please, Michael.
It's fascinating what you said about dose response. Can you tell us a little bit about how you test dose response with the digital therapeutic in a clinical trial?
Yeah, that's a fantastic question here. So the principles that we apply are just PK/PD type analysis, right? So you give graduated doses of the therapeutic, and particularly the mechanism of action. The therapeutic that we develop is a whole patient experience. So it's not that you got clunky boxes, I'm going to be doing this, you're going to be doing that. It's actually a guided or curated experience for that disease and for that person using some matrices that are involved. But its fundamental core is this mechanism of action. So what we do in these early trials, we actually do basket studies. So we use a single intervention across multiple disease states. We also tune how much of that intervention we give, whether it's time on task or how many lessons that you've completed, and then correlate that. We haven't gone all the way to a factorial design, right? Like where we could break out the components and the outcomes there and really get to a PK/PD response. Last, what we do is a lot of real-world evidence, and the real-world evidence actually gives you abbreviated courses or more extensive courses of lessons completed. Does that correlate, right, with outcome data? So that's kind of how we approach it. But great question, exactly. Yeah. Please.
How do you evaluate the patient engagement?
Patient engagement. Yeah, that's like an ongoing discussion here. In fact, this is a safe zone here. I would say that clinical sciences is just as important as engagement science when it comes to digital therapeutics. Remember, taking a pill is a passive act, or having microneedles on your arm, right, is also another passive act. This requires active engagement, or most digital therapeutics. There's some I can show you wavelengths, oscillations that really don't, but most require interactivity, right? So engagement in its crudest sense can be simply app opens, right? And to a more evolved sense as lessons completed, to a more evolved sense as levels of difficulty or tasks, right? That you go into, and to really what the Holy Grail would be is we link all of our studies with actual biomarkers. So we're partnered with, for instance, Altoida. I'm not sure if you're familiar with Altoida. We have a common joint venture with Hikma, and they have an augmented reality computerized testing platform. So what they do is either take the phone or tablet and use it for, let's say I'm an MCI patient or a dementia patient, right? I'm going to claim that a lot of standardized testings are culturally biased, right? You might not enjoy the same foods that are found in your testing, right? Or use the same vocabulary. In fact, in the MOCA exam, who uses the rhinoceros and the lion and the right, and the camel? If you've ever done a MOCA, Montreal Cognitive Assessment, in their daily life, they don't. So it's not ecologically valid. What we do is use tools like Altoida, do it in their own house, so they would see, this is a microphone, I have a bracelet on, I have a shirt on. These are more relevant type of things, reaction times and things of that nature that are kind of correlated to them. So that's one example of how we do it. So biomarkers in all of our studies, so we reduce the ecological invalidity that you have in response there. But then we always go back to the Holy Grail, drug endpoints. So you could use fancy and sophisticated digital or surrogate markers. What really matters for FDA clearance and then also commercial applications is not to be too foreign and not to be too peculiar, right? After all, you're competing. You're not trying to reinvent like a whole new way for doctors to treat and manage diseases. They have already the tools that they're doing over there. You're trying to give them another tool in their armamentarium, not their diagnostics, essentially goes on. So we go after the drug-based endpoints. It's not traits or symptoms or how your self-perception is the disease. If it's depression, it's ambient MADRS, where when I was doing drug research, that's what I would use to get approved. Yeah. Great. Oh, please. Oh, professor, please. Yes. Okay.
So at the gross level, then, how would you summarize the delta effect of the combination therapy over just a standalone therapy?
Yes, I love the question. If a combination is built well, it would actually be synergistic, right? And that's what actually separates this complementary. Complementary is agnostic of what the drug counterpart is. It sits on background medication. You might say, okay, there might be a few classes of drugs for that particular indication. For instance, heart disease, we might say, okay, statins-based therapy, right, for that indication. But once you go into combination, then you could look into the exact profile of the drug acid. And, oh, I love this. This is exactly what I love spending my time on, because then you could see what are the downfalls of the drug acid. Is it having off-site effects? Is it not hitting a sub-item on the FDA-accepted clinical endpoint? Then I would buttress that. I would create, essentially, the digital components to get to that. Now, that's just pure efficacy. Then I could go into the adherence arm. We have a lot of data. When you combine this, right, and you establish a therapeutic alliance that you never get by just taking a blind pill, right, that you have, then you have adherence, which further amplifies efficacy, and then the safety and tolerability. A lot of times you have, there's intended or unintended kind of non-adherence and safety and tolerability issues. You could buttress it. For instance, subcutaneous injections, I could actually acclimate them. That, right, peri-procedural anxiety beforehand, what expectations are if you're going to have a rash or burn, discoloration or bruise, right? All of those type of things. So efficacy, safety, tolerability, adherence in a combination, direct mapping to each other. That's how we get synergy. Yeah. Please.
Your delivery mechanism for any intervention, assuming it's a sub $500 device, what measures or what are your thoughts from a go-to-market strategy to provide equity so that everybody can afford digital therapeutics, not just by carrying a $500 device, which is a big measure or metric for social neutrality?
Yeah, it's a great point. Equity, parity. bridging the divide. And frankly, look, you're hard pressed to find a pharmaceutical company that does not have patient assistance programs and rebates and actually provides a lot of discounted programs there. But we have the potential to go well beyond that. One, the scale. If we're not dealing with biologics and using an incubator and the development costs, right? So the EBITDA margins, we're literally dealing with code and just releasing an access code to our end user for development. Now, to directly address your question, I touched on it a little bit. We don't just design for the latest version. We cover all platforms. We don't, all platforms that are covered by their manufacturer, right? So it's like the 6 or 7 different iOS versions or Android operating system versions that you have. We don't require 24/7 interconnectivity or wide device. We run on Edge. We develop multiple languages. We look at reading comprehension scores. In fact, because we're dealing with multinational companies, internationalization is first to mind. Immediately, when I start a new clinical development program, I'm not just thinking of a US-centric audience. I'm saying, how does this apply to Japan, for instance? So that automatically kind of gets me, thinking on the parity and equity thing. And here, and this is just a lot of speech, so I'll just say, we have a marketed product that is approved by New Mexico Medicaid, excuse me. So Medicaid has very, very strong standards. You need to have languages, right, in their predominant distribution area, the catchment area. You need to have a reading comprehension level that's at the 4th grade status. You need to have troubleshooting capabilities that are all provided there in patient access. You need to support phones that are pretty much accessible under the government assistance packages. On top of that, there's legislation. There's legislation in the works right now. Jean Shane, senator from New Hampshire, is actually one of the co-sponsors on creating this level of mental health parity using prescription digital therapeutics that would actually fund the device itself and the plan itself to even download, right? So data plans and cellular plans to do that. So if that's successful, that would be a good solution in the United States to get this mainstream. But great point. Yeah, this is part of our nucleus and DNA. Yeah, great. Yes.
How are you pursuing reimbursement through traditional payers and formulary positions?
Laura, can we end this session? Yeah. How are we not pursuing reimbursements? Yeah. So here, I'll be brutally honest. So most folks right now are going employer after employer, right? So B2B type of approaches, and we've actually done that in the past. So we have, even though we have some health plans that are covered, we're actually going through employer sponsored benefit programs. Now, in order to get on these types of formularies, there have been headwinds, right? So Massachusetts is probably the pioneer. If you look at MassHealth's non-drug program benefit, you'll see actually 2 digital therapeutics on there, Reset and Reset Health. So it's actually on there. We just have to pave the way for more access. Legislation like this, sponsored by Jean Shaheen, would expand it. There are innovations at the FDA and HHS that requires now them to look at how to support new technologies. CMS, that's I'm talking about, right? They eventually get them coverage that's going on board. But you're right, it's a long drawn out battle. In a nutshell, what I would say is, if I presented to them a data package that was on par or superior to a drug asset, virtually no side effects, no drug-drug interactions, right?
Probably lower cost.
Yeah, lower cost for sure. That's a no-brainer to me. Thus far, that has not happened. So that's why I don't think that there's been a material challenge to the reimbursement structures with a sufficient data and evidence package for this to happen. I'm sorry, I might be demeaning to my colleagues here, but yeah. Jason, okay. Great, we're gonna wrap up. Okay. Yes. I'm gonna hand the mic back. Yes.
Yeah, great job. Great questions all around.
I think we helped everyone quite a bit this evening, but I appreciate you guys staying so late, and a really very grateful appreciation to everyone who made their way this evening for this new event that we're putting together quarterly. to Martin Poster for hosting us and providing us the capabilities to be here this evening to both new technology and be a challenge for all the behind the scenes work to make this all happen. And I hope you guys, especially the young entrepreneurs in the room, have learned and taken insights from industry experts. And for the experts in the room, hopefully there's a renewed energy that you felt from the people presenting here today. So really appreciate it. And Jason,
yeah, I really appreciate everybody coming out, and these are going to be quarterly events. So excited to see all of you back at our events in the future. And yeah, make sure, take some time, make some new connections, and yeah, hope you enjoy the rest of the event here.